All right, guys, today we are talking about cell division. So really quickly, just to backtrack on things that we actually worked on while we were together in school at the very beginning of the semester, we talked about the cell theory and the different scientists who contributed to the formation of the cell theory, but specifically, what does the cell theory tell us? And it tells us that all living things are made out of cells. Cells are the basic unit of life, and you can only get cells from pre-existing cells, so cell division, essentially. Remember that before the cell theory was truly proven, people believed in something called spontaneous generation, which was this idea of getting living materials from non-living materials. And we know that Louis Pasteur was able to really disprove that through his maggots in a jar and rotting meat experiment. And it really showed that you can only get living things from things that were already alive. So again, this process is called cell division. And this is the process of making new daughter cells. So you have two options when it comes to cell division. You can have something called mitosis, and you can have something called meiosis. So sexual cell division versus asexual. So when you're talking about sexual cell division, the goal is to make sperm or egg cells that then would unite together through fertilization to create something called a zygote or a fertilized egg. But when we're talking mitosis, we are talking about asexual reproduction. So think somatic cells, which are a fancy term for body cells. This is the way that your body and living things are able to generate identical daughter cells to the parent. So you would want them to have the exact amount of DNA and genetic material in order to then go through a regular life cycle. So a normal cell goes through reproduction and growth before it actually divides. Think of it as having a life, just like you do. In your unicellular organisms, these lives are a lot more simple than they would be in a multicellular organism. And in your unicellular organism, their reproduction and their cell division tends to be a lot more simple as well. So you're talking one cell that is simply splitting in half to form two exact copies. Bacterial cells do this, yeast cells, but your body cells, somatic cells, will do this as well. Multicellular organisms, you're looking at the purpose being not necessarily reproduction of the cell as in creating a new organism that's going to go off into the world on its own, but instead replacing dead and damaged cells as a part of the whole. So your functions of cell division, again, it can be for reproductive purposes, growth and development, or the renewal of tissues that are dead or dying. Some different types of asexual reproduction. You have binary fission, budding, and mitosis. And then on the sexual side, we'll talk about that later, you have something called meiosis. So quickly to look at those asexual pathways, the first one I want to talk about is binary fission. And this is something you're going to see in bacterial cells. It is asexual. And before the cell can divide, it has to increase itself in size. Because again, you're taking a single cell and you're dividing it in half, essentially, to create two identical cells. So identical cytoplasm, identical organelles, cell membrane, you're going to want to have the DNA, all of that stuff there. And it's going to be a perfect replica of the parent cell, the original cell. So you're going to go ahead and double that DNA, double the components that are there, and then create those two cells. Budding is a different form of asexual reproduction where a projection called a cell projection is actually created off of the organism or the larger cell. So hydra, yeast, things like this might undergo budding. So if you look at the little picture we've got there, you can see your hydra and you can see the little, almost looks like a little arm, but again, it has the tentacles at the top. It's a little miniature hydra, and once it's large enough that it could be self-sustaining, it will then kind of fall off of the parent and it'll be a perfect little genetic copy. Mitosis is where we're going to focus in terms of this asexual cell division, because mitosis is what living things in terms of you and me and animals and plants as well undergo in order to repair and replace the cells that make up their body. We're talking every single cell in your body undergoes mitosis in order for reproduction and replacement, with the exception of sex cells, so sperm and egg. Those are called gametes. So your somatic cells, your body cells, they all use this. The cells that you create through mitosis are going to be identical to the parent. There's something that we call diploid. You'll see this referred to as the number 2 and the letter N, so 2N. That means it has 2 sets of genetic information. So N, think chromosome or genetics, 2N would be 2 sets of it. So you're going to have a complete organism's worth of information in this diploid organism or diploid cell. And there are 4 major phases to mitosis: the prophase, metaphase, anaphase, and telophase. Something I want to highlight for you: mitosis is specifically cell division. Regular cell life is the vast majority of the cell's lifespan, the cell's life cycle. This mitosis piece is a very quick, very short phase within that overarching lifespan. So prophase, metaphase, anaphase, telophase, in the grand scheme of things, they're fairly short. The majority of the cell's life is going to be things like just getting larger and developing and maturing, and then you'll undergo cell division when you go to replace or repair. Before all of this can happen, though, how do we make sure that everything that we need is inside of those cells? And the answer is something called DNA replication. We want to take the DNA that is encoded and exists in the parent organism, the parent cell that we're trying to replace or repair, and we're going to copy it. And we want that copy to be as perfect as we can, because you should know your DNA that you're born with is the DNA you're going to die with. And I say that not in that DNA doesn't have the ability to change between generations, because we know that things like mutations do happen. But when you're undergoing something like mitosis, you're not combining genetics from a parent, a mom, and a dad. You have one parent and you're trying to remake essentially that parent cell. So you're going to want to make sure that your DNA is an exact copy. So how we do that is think of the DNA as being like a zipper. We're going to unzip it. So your DNA helix is that pretty spiral staircase picture you guys have always seen in science classes. And the goal is to take those 2 separate strands and separate them so that we can then use them as a template for pairing up our new DNA. This is called semi-conservative replication. And what it essentially does is make sure that the DNA in the offspring is going to be identical to the parent, because we're using it almost like a stencil. We are making sure that we pair it up exactly like it was in the original. And now we have 2 strands of DNA that are half old, half new. So half from the parent, half that was newly created for the offspring. And that helps us to kind of maintain fidelity and make sure that the DNA is identical and exact to what it was originally. So look at this picture here. You have your parent cell, and you can see that it is dividing into 2 identical daughter cells. They are slightly smaller, but they have all of the same stuff. The organelles, the cytoplasm, the DNA, it's all identical. So then what does DNA look like in cells? And that's not as easy of an answer as it is of a question, because when I ask you what does DNA look like in cells, hopefully most of you are thinking DNA double helix. It looks like that pretty spiral staircase, and that's great. And that is what it looks like if you think about it on its fundamental level, but it has other structures in terms of how it presents itself and how it moves and exists within the cell. So in terms of cell division, there are 3 different forms, and I use forms loosely. I'm doing air quotes even though you can't see me. Forms of DNA that I want you guys to remember for this particular unit: chromatin, chromosomes, and chromatids. So first big thing, the information in the chromatin and the chromosomes and the chromatids, same stuff. That fundamental helix DNA with the base pairs and their different letters, those are all the same. That hasn't changed. We're just talking about the physical form that it's in. So chromatin would be that middle photo that you guys are kind of seeing in your picture here. Chromatin is super loosely coiled DNA that exists in a non-dividing cell. So if I were to show you a picture of a non-dividing cell and you look inside the nucleus, if we're talking about a eukaryotic cell, it would look like what I describe as being a unraveled ball of yarn just chilling. That's your chromatin. Chromatin is super loosely coiled. It is not condensed. That's the term we use to say that it's taken a true like packed shape. It's very loose, almost like limp spaghetti noodle. Okay, that's your chromatin. When the cell goes to divide, it condenses that DNA. It takes it, it wraps it up really tightly. So imagine taking that loose ball of yarn and balling it up into a nice tight coil, and it forms these beautiful, cute little rod X shapes. Those are your chromosomes. So a chromosome has two halves, and those halves are identical. We refer to them as the sister chromatids. So chromatin is the loose stuff, non-dividing cell. Chromosomes tightly coil it up. This happens when the cell starts to divide and undergoes that mitosis process. It's the rod shape that looks like an X, both halves together. The sister chromatids then would be one half of that X shape, and they are joined together by that little yellow dot, which is called the centromere. In a cell that's not dividing, DNA exists as chromatin in the nucleus. We just talked about that. So if you look at the graphic that you have here, you can see your DNA double helix and how it gets wound around. Think about like a thread on a spool, okay? It gets wound around proteins called histone proteins. And then as it gets more and more tightly condensed, that becomes your chromosome. So DNA helix gets kind of loosely wound up to make chromatin. Pull that string-like material really tightly and wrap it up tightly. That becomes the chromosome. Cut the chromosome in half and you've got a chromatid. So again, here's a little cartoon-esque diagram for you. So during cell division, chromatin needs to become chromosomes. So we said that chromatin exists in the nucleus of a non-dividing cell. But when the cell goes to divide, we need our chromosomes to become very distinct because they're going to essentially align themselves and cut themselves perfectly in half and divide so we can ensure that both new daughter cells that we're creating have equal amounts of DNA and they have all of the information they need. This process is called condensation. So when you think condensation, hopefully you think about like water vapor in the air, and when you go outside on a hot summer morning and there's dew on the leaves and on your windshield of your car, that's condensation. So that moisture in the air, it was able to then cool and condense onto the surfaces outside. We use that same terminology of condensation when we're talking about your chromatin becoming chromosomes because it's taking a defined shape, a distinct shape. And again, those are kind of rod shaped. I usually call them X shapes because that's how they're depicted. But if you were to then separate them, they obviously look like little rods. And then your chromatids would be one half of those chromosomes. Again, we call them sisters because they are meant to be a matching set. They're joined at that center point, that centromere, but each sister should have the same information. So something that you might want to kind of keep in your repertoire of cool science information, we've been talking about eukaryotic cells in terms of these DNA structures, but remember that a prokaryotic cell doesn't have a nucleus. Right? So it's not going to have chromatin chilling out inside the nucleus of a non-dividing cell. Instead, its DNA exists in a circular, almost a ring-like shape. Okay? So that circular chromosome that's like attached to the inside cell membrane, that's where your DNA exists within your prokaryotic cell. We call it a plasmid. Okay? There is no nucleus to kind of contain this stuff. Versus your eukaryotic chromosomes, obviously, when the cell is non-dividing, they do exist within that nucleus. So this all then brings us to the cell cycle. And the cell cycle is the life cycle of the cell. So the processes and the phases that a cell goes through during its maturation, its development, and then eventual division. So think about this as being like your life cycle. So fertilization, you're born, baby, toddler, child, adolescent, teenager. You know what I'm saying? There's different phases. Same is true of your cell. So this cute little graphic here you will see on a quiz in a slightly different version, but it's that same picture. So I'm just pointing that out to you now. Cell cycle has two major phases. It has the interphase and mitosis. Interphase is the vast majority of the cell's life. This is where the cell is going through the process of getting larger. It's going through the process of just taking care of its basic needs, finding energy and undergoing all of the organelle processes and just undergoing regular life. Then, as it becomes time for the cell to divide, it will copy its DNA and make all of the structures that it needs in order to successfully undergo mitosis. So if you look at your picture, that is the greenish blue arrow you're seeing going around like 90% of that circle. And then you have the little yellow arrows. That's the mitotic phase or mitosis. That is where the cell is actually physically dividing itself. Okay, and then you can also see a tiny little sliver called cytokinesis. So cytokinesis is this division of the cytoplasm. Mitosis before that is actually called karyokinesis because it's the process of dividing the genetic information, like the nuclear information that's inside of it. So I'm not asking you to know the word karyokinesis. I'm just throwing that out there for funsies. But interphase, vast majority. Mitosis, very, very small sliver, where the cell is now going to divide into a daughter cells. So in my interphase, we talked about it. It's growth and replication. Mitosis is cell division. But within my interphase, there are 3 phases. So interphase is the collective term, but within interphase, we have G1, S, and G2. So your G1 phase stands for growth 1. And growth 1 is normal cell life. Okay, the cell's getting bigger, the cell is maturing, the cell is going about its daily tasks and routines as it would, and it can live in this stage indefinitely. So not all cells have to divide. If a cell is in an environment where it is not necessarily thriving to a point where it can dedicate energy and resources to the process of division, it will stay in growth 1. It will not leave this phase if it doesn't feel like it would successfully survive it. Because once you leave growth 1, you go to the S phase. And the S phase stands for synthesis. This is where DNA replication happens. And this is where the cell becomes fated to undergoing the process of mitosis. Once you go to the S phase, you're doubling your DNA with the intentions of then dividing it so that you can create 2 daughter cells. Once you've doubled that DNA, you can't go back from that. You have to undergo division because the cell can't successfully survive with extra DNA. The cell has the exact amount that it needs, that diploid, that 2N. Once you double that, you have to undergo division, otherwise the cell would die. After you go through synthesis, you're going to go to something called G2. This is a secondary growth phase, but instead of being growth as in regular like cell maturation and development, this is growth specifically with the intention of division. So doubling all of the cellular components with the intention of giving part of it to the different daughter cells. So doubling the cytoplasm, making sure you have 2 sets of all the organelles or whatever it might be. So you're creating the things that you need for cell division. And then you're going to hit mitosis, that little yellow phase. So this is that final stage of the cell's life cycle, because beyond this point, one cell is going to become 2 daughter cells, and it'll go back into the whole phase as interphase again. So we talked about the 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. Easiest way to remember them is the word PMAT. If you can remember PMAT, you can remember the different phases: prophase, metaphase, anaphase, telophase. So if you kind of look at this sketch of the cell cycle again, you can see the blue is the interphase. It's vastly larger. This one is a little bit less correctly drawn as maybe the last one. But you can see the interphase is the vast majority. You have your G1, cells mature. S, DNA is copied. G2, cells prepare for division, and then undergoing your mitosis phase. So prophase, metaphase, anaphase, telophase, and then eventually cytokinesis, which is where you fully split, like cut in half into 2 daughter cells. I want to go through the different phases of mitosis. I need you to know, 1, what do those phases look like in a cell? And then 2, what happens distinctly in each phase? Because they have very distinct steps, which allows us to classify based on an image of a cell exactly what is happening and exactly what part of mitosis that cell is in. So the first one is prophase. And prophase is the preparation phase in terms of getting ready for division. So I don't mean getting ready as in like growing like you would have in interphase. This is where you're creating the different components that you need specifically within mitosis. So you're going to make sure you have chromosomes. So turn the chromatin into chromosomes, and you need to disintegrate the nuclear membrane. So the chromatin exists inside the nucleus, and we need to get that out so that those chromosomes can be kind of loose within the cell. So you have to destroy the nuclear membrane, you have to condense the chromatin into chromosomes, make those distinct rod X shapes, and you need to form something called the spindle apparatus. And the spindle apparatus is a structure and network of microtubules that is formed from components of the cytoskeleton and allows for almost like a framework for the chromosomes to move on easily during mitosis. So here's a closer picture of that spindle apparatus for you, and you can see that the chromosomes are using it almost like a highway, a network of fibers that they can move on. So if you look at this picture here, you've got interphase transitioning into the prophase. So you have the nucleus with all of the chromatin inside of it, and then eventually that nuclear membrane will disintegrate and that chromatin will coil up to form chromosomes. So here is a picture, an actual cell slide image of what prophase would look like. And what I want you to notice is that you can see the chromosomes where things are starting to become condensed. Like you can see the space in between the different structures. And also, if you look around the nuclear area, there is no solid, thick nuclear membrane line around it. So that is kind of your indications of prophase there. Metaphase would be your second phase. Meta means middle. Okay, so keep your M's together when you're thinking about this. The metaphase is where all of the chromosomes line up in the middle of the cell, in the middle of the spindle. We call it the equatorial plate. So think equator of the Earth, and the poles will be the north and south pole. It's the same idea when you're thinking about the cell. So those chromosomes are going to line up at the equator, and they're going to be attached to those spindle fibers. So again, a little diagram, and it's showing you that they're moving to where they're lined up in the equatorial plate. So here's a cell picture of it. Same thing. If you're looking at the cell, notice there's no nucleus anymore. You can see the distinct chromosome black shapes, and you can see where they're clearly lining themselves up. Here's another option for you. You can see the spindle really well in this image, which I like, and you can see the distinct poles. So obviously those chromosomes are lined up very centrally, and then in our next phase, which is anaphase, they're going to separate. So anaphase is where you go from having chromosomes to having what we call the sister chromatids, because the chromosome has separated at the centromere, and now you have two identical halves. And each half is going to go to a different pole. So half goes to the North Pole, half goes to the South Pole, and creates a division, an empty spot in between the two, which is eventually where the cell is going to cut itself in half. So anaphase, I always tell people the easiest way to remember anaphase is that Anna is a girl's name, and people have the ability to walk around. And anaphase is the phase where you're seeing these sister chromatins essentially walking or moving towards the poles. It's how I remember it. I know it's kind of cheesy. So again, here's an image of that for you guys. You can see there is a very distinct space between the two, and you can see the chromosomes moving towards the edge. And then lastly is telophase. Telophase is the reverse of prophase. So this is where all the things that you undid or created during prophase, you have to go back on that. Cells can't sustain themselves long term without their nucleus. So you're going to reform the nuclear membrane. You're going to take the chromosomes and you're going to uncondense them now back into chromatin form. Put it back in that loose kind of spaghetti-like form. And you're then going to have the spindle go away. Spindle apparatus falls apart. Those microtubules go back to their regular daily lives. And then cytokinesis takes place. So if you look at this diagram, this picture, you can see where it almost looks like two googly eyes, essentially, where there's a pinch off towards the center. That's called the cleavage furrow. And the cleavage furrow is the location where the cytoplasm is beginning to pinch itself separate. So telophase is where the cleavage furrow forms. And then this is immediately followed by cytokinesis. So during telophase, we have now officially had our genetic information divided. Now with cytokinesis, all we have to do is divide the cytoplasm and reform the cell membrane on each side. So now you've gone from having the materials for two distinct cells, but they're still kind of held together by cytoplasm, to two completely separated cells. So here's your telophase, however you want to call it, image. And again, a picture of it in terms of what it would look like in an actual cell slide. So you will find, if you were to look at cell slides in class, which we would have done had we still been in school together, people tend to struggle with telophase versus anaphase. And what they struggle with is it's not always super clear with that line in between them. It depends on like when during telophase we're talking and how close to cytokinesis we actually are. But what you will actually see that can help differentiate that for you is the pinching. Are you seeing the pinching of the cell off, and are the chromosomes actually at the pole as opposed to moving towards it? Then lastly, this little slide section here is going to just show you the different interphase through cytokinesis, just to kind of show you what's happening, almost like a time lapse if you go through that. Okay, so 4 phases of mitosis. Take a look at these pictures. Take a second by yourself and see if you can match them up. Which one is prophase, metaphase, anaphase, and telophase? And hopefully you found prophase as being that one right there. So you can see the nuclear membrane is disintegrating. You can start to see those fibers from the spindle apparatus beginning to form, and then you are seeing the condensation of those chromosomes. For your metaphase, meta means middle, they're lining up there at that equator. Anaphase, they're starting to migrate to the poles. And then lastly, your telophase, you're starting to see 2 distinct cell shapes. And this brings us to meiosis. Meiosis and mitosis are very, very similar, except for mitosis is asexual. Meiosis is sexual. So meiosis is for creating sex cells, sperm, egg. We call them gametes. So instead of making 2 cells like you did during mitosis, you're now making 4 cells. And the reason for that, and bear with me, like think this through with me, when you're talking mitosis, if you're making a skin cell, your parents didn't have to have sex every time that you create a new piece of skin, okay? It already has diploid genetic information. It has all of the genetic information that it needs. Whereas sperm and egg, the goal would be for those 2 cells to come together to create the offspring. So if the overall goal is to end up with diploid, you would need something called haploid. So think half. Haploid is 1N. So 1N plus 1N gives you 2N. See where I'm going with this? So instead of making 2 cells that are 2N, we're going to make 4 cells that are 1N, so that when we put 1N and 1N together, sperm with egg, we get a baby that is 2N. Okay? So meiosis and mitosis, same processes as in the phases that they go through, except for meiosis is going to go through it twice. So whereas you'd have prophase, metaphase, anaphase, telophase for mitosis, with meiosis, you're going to have prophase 1, metaphase 1, anaphase 1, and then you're going to go through it prophase 2, metaphase 2. Okay? So 8 stages now. So here's a diagram that explains it a lot better than I probably just did. So you have your interphase. When interphase is done, you have doubled your DNA and you're ready to go through division. You go through like you would for mitosis, prophase, metaphase, anaphase, telophase. Now you have 2 cells that are 2N. But I'm not going to stop there. I'm going to divide those cells a second time so that now there's only 1N. So prophase, metaphase, anaphase, telophase again. Okay? So then how are they different? Obviously the number of cells that are made. Mitosis is making 2, meiosis is making 4. The kind of genetic information or the amount of genetic information in those cells. Mitosis is making diploid, 2N. Meiosis is making haploid, 1N, because body cells need to have 2 sets of genetic information. Sex cells only need 1 because you're going to add the mother's information with the father's in order to make the offspring. Mitosis has 4 phases, meiosis has 8 phases, and then obviously they're making different types of cells. Somatic cells for mitosis, gametes for meiosis. Okay, so if you look at your mitosis here, you're 46, and then you're dividing down to identical genetic information. Meiosis, we're going into 23 chromosomes as opposed to 46 chromosomes, because the idea is you add your mom's chromosomes with your dad's chromosomes, and that gives you the child. So then why is meiosis necessary? Because it's the fundamental basis of reproduction. In order to have sexual reproduction, we have to have the ability of creating an offspring using information from both parents. And if we didn't have haploid sex cells that are created through meiosis, we would end up with babies that have too much genetic information and they won't survive. So this kind of creates the foundation necessary for us to create these sexually unique organisms through sexual reproduction because of the ability to create haploid cells. Okay, so fertilization is like putting it all together. You take haploid plus haploid and you get diploid again. 1N plus 1N gives you 2N. And there's your little equation, okay? Last slide. Again, it's just showing you information from mom plus information from dad. If it wasn't haploid, if you tried to take diploid cells made through mitosis and put them together through sexual reproduction, you would get a child with too much genetic information. But if you use meiosis to create a haploid cell, which is your sperm and your egg, and you add those together, you're able to create an offspring that has the perfect amount of DNA.